Today is a very special lecture. This is the 22nd webinar of the Ocular Genetics Interest Group. But today is 28th February, which is celebrated as the Rare Disease Awareness Day. And so we thought to keep a special lecture from Dr. Rob Hufnagel. He is a close friend and kind of mentor for me during my initial time at the Ocular Genetics, at the Global Eye Genetic Consortium. And he's also a good friend advisor and has helped me with some of my cases and my problems during managing some of my while managing some of my cases. It's and we are very thankful to him for joining us. It's around 5 a.m. For his time in Hawaii. And we are so grateful that he agreed for this lecture and making this Rare Disease Day special for all of us. Rare Disease Day is celebrated at the end of February, the last day of February. So on the leap year, it is 29th February, and the rest of the years, it is 28th of February, to create awareness about patients suffering from rare diseases, which are around 7,000 plus diseases. And as we all know that these diseases are quite neglected in day-to-day practice, not just because of the rarity, because the clinical management is also not very clear or clear cut for many of them. So they end up even diagnosed being late. So this webinar is also uh, a kind of effort from us to create awareness for these diseases. And Dr. Hufnagel's topic is also very apt for it, which where he'll be talking about the precision medicine in rare disease or ocular genetic diseases. So I'll just start with a brief introduction. I'm saying it brief because actually, if I introduce him all of his accolades, then it'll be quite a long talk of mine only. So, as I mentioned, this is the Genetics Club 22, and the topic for his talk is Enabling Precision Medicine in Genomics Era. Dr. Hufnagel is a physician scientist specializing in clinical ophthalmic genetics. He joined the Center for Integrated Healthcare Research at the Kaiser Permanente in Hawaii in 2023. Um, he's a rare combination of being a pediatrician, then a geneticist, and then joining ophthalmic genetics at the NEI and NIH, and then also at the clinical and molecular genetics and genomics at the NHGRI, which is the National Human Genome Research Institute. So multiple degrees and multiple fellowships to him. He is currently the director of the molecular genetics at the ACMG board of directors. He is also chairing the clinical domain of the working group for ClinGen, which is Clinical Genome Resource. He serves on the scientific advisory board for the Foundation for Fighting Blindness and editorial board for ophthalmic genetics. He has over 150 peer-reviewed publications, primarily all of them in ophthalmic genetics, especially retinal dystrophies, genomic diagnosis. So all of these things, the NEI, the NHGRI, CMG, ClinGen, all of these are things that we kind of read about and we look up to when we are, embarking on the journey of ophthalmic genetics. And Rob has been at the center of all this. So it's an honor to be a part of, witnessing his lecture today on the occasion of Rare Disease Day. So, Rob, if you're ready, then we can start with your lecture. I think he's reconnecting. There's some technical problem. Sorry for the inconvenience and kindly bear with us. Meanwhile, if anyone wants to share how Rare Disease Day is celebrated at your institute, or if you do something about it, like in at Shroffs, we participated in the organization for rare disease, the uh, the ORDI India. It conducts a race which was on 22nd of February this year, last Sunday. So it's a symbolic race, 7 kilometers or 14 kilometers, called Race for Seven, to create awareness for the 7,000 rare diseases which are present. It's a fun event, at the same time creating awareness, and you run to support the rare disease that you support. Apart from that, we are having this lecture. So any other special things has been done at any places, then please share with us. Meanwhile, Rob is getting connected.
Hello.
Hello. Yes, ma'am.
Yeah. So in at Kaminini, because we are a medical college, we
Yes, ma'am.
try to create awareness to the undergraduate students so that from their medical education itself, they get aware that they have to keep their eyes open to look for rare diseases so that the diagnosis is not delayed so much. Uh, we talk to OBGs, then pediatrics, as well as undergraduates. And
Mm-hmm.
also had made Instagram reels to small ones where we have given how best we can uh, look at look at different diseases, and with the 3 or 4 questions told them that what sort of testing may be required, so that people who follow the Instagram page can get. So we took one oncology case, one obstetric case, and so on. So that's what we did.
Yes, ma'am, that's a very important way to look at it and deal with it, because one has to start from the basics. I remember when we were students, the genetics chapter was kind of skipped. There was no genetic chapter also, and we tend to skip the gene names and everything when we were studying, because we didn't think it's important, and never it was stressed that it is important. So that's the way to go and begin with the undergrads. Oh, thank you, ma'am. So Dr. Hufnagel is here with us. Good morning. You need to unmute yourself.
You so much. I'm sorry about that. My Zoom decided it needed to reinstall the new version as soon as I tried to join. My
Oh,
apologies.
oh, that's okay. And we are again, I don't know how much you heard about your introduction, but we are really thankful to you for, taking out time, especially at this odd hour. And because of the time difference, I think these are the only two times that are possible, either too late for us or
It's
too
fine.
early
It's fine.
morning.
It's my fault. My
So,
fault.
but we really, really wanted to listen and hear from you. So, without much more, taking more time, please go ahead with your lecture, Dr. Abnagal.
Thank you. Thank you. And again, apologies for my technical difficulties. Do you see my slides, the full slides?
Yes,
Okay,
yes.
fantastic. All right. My name is Rob Hufnagel. I've been on the OGIC chat, I believe, since Dr. Shelja started it, and so it's been wonderful seeing all the wonderful discussions, comments, and seminars that people have had, and it's really my honor to be able to give this special one, and I'm looking forward to interacting with everybody today. Just to go into, I was talking with Dr. Shalja about what we should talk about today, and so this actually nicely sums up what I'm going to be covering: an intriguing case and mysterious unsolved cases, syndromic dilemmas, and I hope getting into fundamental genetics. In my career path, I did my MD PhD at the University of Cincinnati, and my post training was pretty circuitous. I knew I wanted to do ophthalmology. I knew I wanted to do genetics, but I wasn't sure which way to get there. And so I eventually decided to go into pediatrics and medical genetics and then specialize in ophthalmic genetics from the medical genetics specialty. And I had the great opportunity to work with Dr. Brian Brooks, an ophthalmologist and medical geneticist, double boarded at the National Eye Institute, and several other ophthalmic geneticists, and also had the opportunity to mentor Dr. Shelja when she was visiting there as well. And so the clinical molecular genetics was also completed at the National Human Genome Research Institute. So I really have been doing my research as a partnership between ophthalmology and genetics to provide people with answers for their genetic conditions. And I currently work at Kaiser Permanente out here in Hawaii, looking at how we use genetics at point of care. In our first case presentation, be talking about this 8-year-old young woman who was referred for diagnostic evaluation of syndromic retinal degeneration, previously diagnosed with Leber congenital amaurosis, early onset retinitis pigmentosa, and choroideremia. She carried many different diagnoses depending on who she would see, and her brother was similarly affected. By 6 years, you can see her visual acuity there was starting to diminish, and she failed a screen from her pediatrician and was referred by an optometrist, and then eventually to the retina specialist. But even over the course of a year and a half, you can see that her vision decreased dramatically from 20/60 to 2800 in the right eye, and it was not improved with glasses. And interestingly, she was born at 37 weeks, normal birth, some hyperthyroidism detected at birth. It was treated. This is covered in our newborn screening program. And she also had growth hormone deficiency on further endocrine workup. And then as an infant, she had these transient developmental delays that were pretty global through 6 months. All of her milestones, gross motor, fine motor, were all delayed, but they were resolved over time. And on brain MRI, eventually, as part of this endocrine workup, she had a small anterior pituitary. And here we can see her eyelashes. You can see two things. One, she has very long and bushy eyelashes, and they're very darkly colored, and yet her scalp hair is very lightly colored. You can even see a gray hair here. She also has a very small pituitary, as noted in the brain MRI. And when we look at her retina, you can see just frank atrophy across the retina. And you can imagine why she had all those diagnoses. She's got frank panretinal atrophy, and she also has these scallop lesions that you also see in choroideremia. So she has full thickness atrophy of the retina, RPE, and even the choroid layer. Affecting both the neural retina and the pigment tissues. And then on the nasal side, she also has, this is her brother below, and her brother has a lot more pigment accumulation than she does. But they both have these islands of coalesced pigment here too. And then on the fundus autofluorescence, you can likewise see that there's a lot of hyperfluorescence around the optic nerve, but then a lot of hypoautofluorescent patches showing the RPE dropout. OCT fixation was very difficult for both young people, and so the fixation quality is not great, but what you can appreciate is that there's a lot of retinal thinning, especially of the outer layers and the RPE, and you also see a good bit of retinal remodeling here around the foveal center. And these are Goldman visual fields. Their Goldman visual fields were both highly constricted, both at the 1,3E, 3,4E, and 5,4E. ERGs were severely reduced, both scotopically and photopically. And so this is a very interesting clinical diagnosis where an individual has hypopituitarism. So remember, she had hypothyroidism, growth hormone deficiency, and small pituitary, trichomegaly, and chorioretinopathy. And there had been 14 patients reported since 1965. And this was in the mid-2000s when I saw them, and there's no genetic cause. But we had a family here where we could really start to study this genetic condition. And when we did whole exome analysis, when we send exome on you, me, anybody else, there's always about 100,000 changes against the reference genome. And we used some pretty early systems to study both a homozygous and a compound heterozygous model for her and her brother and her two parents, with the assumption that the parents are carriers for the variants that the patients have. But we found this variant in PNPLA6, these two missense changes, and PNPLA6 had actually only been associated with an adult onset spastic paraplegia type 39, and so we were stumped a little bit. We were thinking, well, maybe we've diagnosed them with an adult onset condition, this spastic paraplegia that they don't have, and they had very normal neurologic exams. So we kept thinking about this variant. We couldn't find any other variants in the list that were of interest and that would fit an autosomal recessive model. And so, we kept going to try to find additional patients and try to understand what might be going on. In our second case presentation, this is a 17-year-old referred for nystagmus and tremor, very different history than our siblings. The vision loss started at age 6 with nyctalopia, central and peripheral impairment, and then hand tremor at age 11, so predominantly neurologic symptoms. And with head bobbing and clumsiness. And this is a picture of his retina. You can see first that the retina, that this person has more ocular pigmentation than the siblings. But there's also this frank atrophy with these coalesced retinal spots, and more of a yellow pigment in the macula. But you can also see some of the fibrotic changes here and severe atrophy in the left eye. And the vision was quite low at this age. And then on the fundus autofluorescence, both near and wide field, you can see the RPE dropout, but also those sort of scallopy looking lesions, this sort of curvilinear dropout areas of RPE surrounded by areas of mixed hyper and hypoautofluorescent RPE, showing the poor health of the retinal pigment epithelium there. And likewise, in his retinal exam, we found that there was retinal thinning, remodeling, and these pigment depositions of material underneath the fovea layer of the RPE. And this is a cut through that fibrotic and was apparently atrophic change, and you can see that there are increases in those subretinal deposits around the RPE. Here on the OCT slice. So his history was limited. He lived in Ghana with his mother until about a year before seeing us. Again, had tremor and ataxia, delayed puberty, and short stature. So different presentation and a later presentation than the two siblings we talked about in the first case. He did have a low LH hormone and low normal FSH. These are the hormones involved with pituitary changes, which would explain his delayed puberty. And these are, of course, pituitary secreted hormones. And but the rest of his workup was pretty normal, including an ataxia gene panel. And this combination of diagnosis is also very interesting. So this is retinal degeneration, hypogonadotropic hypogonadism, and cerebellar ataxia. And it turns out that around the same time, this syndrome called Bucher-Neuhauser syndrome, that there was a paper that came out indicating that PNPLA6 may cause this condition with essentially cerebellar ataxia, and which was similar to the spastic paraplegia that the PNPLA6 had been previously associated with. These pituitary findings in the retinal degeneration. So we thought that we were onto something. And so we did genetic testing for this person, and we found that he had homozygous variants in PNPLA6, this Lu 1051 SER, but it hadn't been characterized yet, nor had the missense variants we had detected in the siblings in the first case. And then our third case presentation, this was a 30-year-old male who's referred to us from NINDS. They wanted him to get a retinal exam because he had spastic paraplegia, and they were thinking that he might have this PNPLA6 disease. But at 3 years old, he had balance issues and walking delays, generalized hyporeflexia by school age. By 22 to 25, he was using a walker. And then he was diagnosed with dystonia and spastic paraplegia. But overall, he had normal height, growth trajectory, no issues with his pituitary, no issues with his vision. And indeed, when we saw him, we did not find anything on dialyte examination, nor did we find anything on the fundus autofluorescence, the ERG, or the visual fields. But we did diagnose him with spastic paraplegia type 39, which was the disease that was originally described with PNPLA6, and found novel homozygous variant in him. So we have all these variants of uncertain significance in PNPLA6, and we have these patients whom we think have PNPLA6 related disorders, but they're very drastically different in their presentation. And so to summarize this here, here's our patient. So 1 and 1.5 are the siblings. 2 is the second case with Bucher-Neuhauser syndrome, and 3 is the case who has normal vision and spastic paraplegia type 39. And you can see our first 3 patients had that really striking chorioretinopathy. The first 2 had congenital hypopituitarism, and the third had an adolescent onset hypopituitarism termed hypogonadotropic hypogonadism. And then the second and third cases both had the spinocerebellar ataxia and spastic paraplegia. And then here we can see the variants, which we all notice are between the 1,000th amino acid up to the 1,300. So we thought, well, there's this interesting cluster of variants here. So let's try to understand this a little bit better. So talking about the diseases, Oliver McFarlane syndrome was first described in 1965 by a group at Moorfields, and so this is by Dr. Oliver and Dr. McFarlane. You can see that the very first case reported had very, very long eyelashes, very, very bushy eyebrows, but a very similar retinopathy. And you can see here that our patients here, and adding another patient here. This is another patient that we had recruited for a larger study I'll talk about, but had recruited him from the UK. And you can see they do have long eyelashes, but not to the same extent as the original published person here. And so we were thinking, well, this is a good diagnosis. There's a good hypothesis that this gene PNPLA6 causes Oliver McFarlane. And what we did notice is when we put a lot of patients together and we recruited almost 20 patients with this condition, that we found that many had long eyelashes, many had alopecia and premature graying, especially for the early onset severe form. And then those with neurologic symptoms, which didn't always happen, hypopituitarism was the most common. But we also saw individuals with spastic paraplegia, cerebellar atrophy, and peripheral neuropathy and muscle wasting. And the chorioretinal degeneration was pretty typical, but of course with a broad presentation based on the age of the person and the age of onset. And we saw various levels of pigment clumping, which did differ from person to person. So to summarize the PNPLA6 disease spectrum and our hypothesis that this was really causing 3 distinct diseases that were overlapping in their nature, we have spastic paraplegia type 39 that was described in 2008. This is an ataxia and cerebellar atrophy, spastic paraplegia, and peripheral neuropathy. Gordon Holmes and that Bucher-Neuhauser syndrome I mentioned were published in 2014, and this was the combination of ataxia, cerebellar atrophy, hypogonadotropic hypogonadism, and chorioretinal degeneration. And then finally, Oliver McFarlane syndrome and a related Lawrence-Moon syndrome that we described in 2015 with ataxia, cerebellar atrophy, and then this really striking congenital pituitary phenotype, the chorioretinopathy, as we've been discussing, and these hair anomalies. So are these 3 different diseases caused by the same gene, or might these be called a spectrum? But I do want to take 1 minute to talk about Lawrence-Moon syndrome, because Lawrence-Moon syndrome we often learn as part of Bardet-Biedl syndrome, or they call it Lawrence-Moon Bardet-Biedl syndrome, formally. But what we found when we went back to the literature was that when we look at Oliver McFarlane in the original report, and then Lawrence Moon in the report in 1866, which is in French, and then the original Bardet-Biedl reports, which were from 1920 to 1922, they have some really interesting overlap with our Oliver McFarlane syndrome, and that while they don't describe the hair phenotypes, there is intellectual disability and retinal degeneration. Notably, the choroidal atrophy was reported in the first Lawrence Moon case, but not in the first Bardet-Biedl cases. And the Lawrence Moon cases also indicated anterior hypopituitarism and short stature, and that the intellectual disability was due to the hypothyroidism, also known as cretinism. But that Lawrence Moon syndrome was potentially actually a case of a different syndrome than Bardet-Biedl syndrome because of these differences. And then furthermore, besides that the Lawrence Moon syndrome has these pituitary phenotypes and the central neurologic phenotypes where Bardet-Biedl does not, when we look at Lawrence Moon, they also don't have obesity and polydactyly, whereas this is very common in Bardet-Biedl syndrome. So we also hypothesize that Lawrence Moon syndrome is actually more similar to Oliver McFarlane syndrome, and that many of these cases may be caused by PNPLA6, and that's what we've also shown. But what's so interesting about this protein? How could PNPLA6 cause 3 different diseases in different age ranges and also with different phenotypes across the body? And you see that kind of variable expressivity where you have such a big spectrum in age and tissue onset and progression. You often see that with enzymes. And so that's actually what PNPLA6 encodes. And the reason that PNPLA6 was discovered wasn't by genetics and the Genome Project. It was discovered in the 1930s as a result of toxicology studies. So in the U.S. and many places in the world, in the 1930s, there were a lot of unsafe substances that were placed into drinks, and so our early versions of Coca-Cola here actually contained cocaine because that was a stimulant that would give people energy. But of course, cocaine is heavily addicted, and that was discovered, and then it was removed from Coca-Cola. Likewise, a lot of alcoholic drinks would contain organophosphates, because organophosphates work in a similar way, suppressing the parasympathetic nervous system and giving a sort of a high energy sympathetic-like response. And so this Walker's Jamaica Ginger was a drink that actually contained organophosphates. And so there was advertised as giving people a lot of energy. But what they found was that people had this wasting disease when they were taking this tonic too much. And people were eventually jailed for essentially poisoning with organophosphates, which are recognized as a poison. But they were jailed because they had added this to the drink to make it more, to give that energy feeling to people. But it was also, of course, like harming people and leading to early deaths. And it also opened up a new form of organophosphate poisoning. So there's inhibition of cholinesterase or acetylcholinesterase, which is what we classically learn in medical school. But there's a second form of organophosphate poisoning called inhibition of neuropathic esterase, or NTE. And this is a Wallerian type degeneration of the CNS and PNS. And what happens is that you get this delayed neuropathy due to the organophosphate poisoning, which is different than the kind of acute cholinergic syndrome that we learn about when people are exposed to farm fertilizer chemicals and things like that. This results in this OPIDN through the inhibition of NTE. Well, it turns out that the PNPLA6 gene encodes this NTE enzyme, this neuropathic esterase. And so this maintains a phospholipid homeostasis and choline recycling. in all cells of the body, and it utilizes phosphatidylcholine and lysophosphatidyl which are part of our our membrane lipid bilayer of all membranes in our bodies, as well as phosphatidylethanolamin and phosphatidylglycerol. And so it functions as a phospholipase A2 in that it basically will remove two of these esterase groups and replace them with hydroxyl groups. And so this is the NTE pathway. And this is the intermediate along the bottom. And it's also important in choline recycling. And so a lot of these products then go on to generate choline, which is important for neurotransmitters and other second messengers. So along the lines of mentoring, I had the opportunity to mentor a lot of wonderful people, including Dr. Shalja. And my graduate student at the time, Jimmy Liu, he really wanted to understand this because he wanted to answer this basic question of, are these really different diseases, or are they actually one disease along the same spectrum? And what we had to do to demonstrate that was to understand whether these genes all affect, or these variants causing these different diseases, could we predict what disease each variant would cause based on how it would affect the enzyme? And could we actually show a dose response type of curve where if you inhibit neuropathy target esterase to a certain extent, can you elicit more and more of these phenotypes in a tissue specific manner? And is that why we have all these different disease names, but they're really all just one disease on a very massive spectrum? And so what we first did was he did a really deep literature dive and looked at phenotype onset in PNPLA6 disorders across different tissues. So from the eyes, from the endocrine system, from the central nervous system, and the peripheral nervous system. And what he found was pretty curious, that while a lot of times the clinicians would define a disease by the age of onset. So for instance, Oliver McFarland syndrome, it's defined by a congenital hypopituitarism. And so we see people are pretty accurate about putting people with the congenital form in that diagnosis. There are also people reported who have a later onset form of anterior hypopituitarism that actually overlaps with Bucher-Neuhauser and Gordon-Holmes syndromes. Likewise, with the ophthalmic manifestations, we found that yes, while the severe form, Oliver McFarland syndrome, was often reported in the literature as being very early onset, there was still plenty of overlap with people who had Bucher-Neuhauser syndrome. And this is also lends credence to that this could be a spectrum. And then central and peripheral nervous system onsets were essentially the same across all of these disease categories. So perhaps these names aren't really distinct diseases, but they are ways of describing the manifestations of PNPLA6. So next, he did a very onerous task of he took all of the variants that were reported in the literature and he looked to see how they clustered. And what he found was that this is the NES domain. So this is the enzymatic center of the protein. These CNB domains are the powerhouse of the enzyme. So they're where ATP and GTP bind to power the enzymatic center. And then this TMD is a transmembrane domain because it sits at the endoplasmic reticulum. And so when we look at this, distribution, we find that about 40% of them are in the NES domain, and then the others are spread throughout the protein. But that about a third in black here are truncating alleles. So they're splicing, frame shift, or nonsense. And that the proportion here across diseases, though, was not even. What we found is for the earlier onset conditions and the ones that had retinopathy and pituitary issues, they actually had a higher likelihood of having the mutation within the enzymatic center, whereas the ones with more adult onset that don't have retinopathy, that they are their mutation, they had a lower proportion of mutations in the NES center, and some of them were outside of that enzymatic center. And what that said to us is that, well, maybe there's a different sensitivity that any disruption of PNPLA6 is likely to lead to issues with age in the central and peripheral nervous system. But for pituitary and retinal onset, you have to have a further reduction of enzyme activity in order to have disease. So could this activity level be really a key driver in disease pathogenesis in different tissues? And so to put this in hypothesis form, that Jimmy wanted to test whether residual activity predicts the tissues affected and the disease severity. And so we did this experiment where we would transfect each of these different mutations, and he tested over 50 mutations and transfected them into cells, into immortalized cells. And then we lysed the cells and then did a colorimetric enzymatic assay that would give us, under a plate reader, would give us the level of activity. And this had been published along the way when neuropathy target esterase had been studied for its toxic effects. Somebody had made a really nice assay for studying the residual levels of the NTE protein as a way of study for toxicology studies. But we're now using that for genetic studies and for diagnostic studies for people. So it's really nice to see how that can come along from the 1970s all the way into the present. And then we wanted to really understand that biological relevance. Why do some patients have central nervous system issues, and some have retinopathy, and some have pituitary, but not all? And that hypothesis that the retina and the pituitary require a further reduction of NT activity was the hypothesis. So these are all the variants that he looked at. And you can see they're going from the N terminus to the C terminus, and then we put all of the null alleles here at the end. And you'll notice a few things. One is that they are of different levels. And what we found was that when we looked at this as the activity relative to wild type, so we always included controls, we found a few things. One is that all of the nulls were nulls, except for variants that were out very distal to the enzymatic domain. So these two frame shifts at 1305 and 1361 are over here in the C terminus, and this often escapes nonsense-mediated decay, so they aren't full nulls. And what we found was, yes, there's actually preserved enzyme activity out here when you have these truncating mutations. And so these people can have milder disease if you have a very distant truncating allele. And the other was that, we would see that these different variants had different levels, and a lot of times that just depended on the severity of the amino acid change, but that most of them in the enzyme domain did have a activity that was lower than 50%, but a few were above 50% there too. When we look at our cases, these are the kids within our first case. This is one of the variants that they had. And then this was the variant that we found in the second person. And this is the variant that we found in the third person in our case series. And so what this is also showing is that these were the people who had the most severe disease, our first case, and the person who had the more adolescent onset in the second case, and the adult onset without retinal degeneration or pituitary issues. So we wanted to really do this and say, okay, well, these first three cases that we saw, we were able to say there's an interesting correlation here, but we wanted to really do that statistically. And so what we did was we estimated residual NTE activity in the PNPLA6 disorders by taking this enzymatic assay combination and then reconstructing patient genotypes. So people either have homozygous or compound homozygous findings, variants. And so we can estimate their residual enzyme activity by taking the average of the two alleles. So in this case, the people in the first case had an estimated NT activity of 23%. And then we can look at that across all of the different categories of disease. And what we found was, indeed, the people who were diagnosed with Oliver McFarlane syndrome tended to have a lower enzyme activity, but when we look and compare it to Bucher-Neuhauser syndrome, there's actually no significant difference. That they were, while you might have more clustering a little bit lower, those with Bucher-Neuhauser or Oliver-McFarlane syndrome as a cohort actually had similar enzyme levels, but both were lower than those who had SPG39, the adult onset that does not have pituitary or retinal findings. So then we said, okay, let's ignore the labels that we put on people. We've labeled people with Oliver McFarlane, Bucher-Neuhauser through the literature. But let's just say who has retinopathy and who does not have retinopathy, who has pituitary disease and who does not have pituitary disease, and ignore what their clinical diagnosis was, Bucher-Neuhauser, Oliver McFarlane, or so on. And so what we found was that those with retinopathy indeed have significantly lower enzyme activity than those who do not have retinopathy, and that those who have endocrinopathy and the pituitary dysfunction did have lower enzyme activity than those without endocrinopathy. And it's interesting then because it suggests that maybe there's a threshold that above a certain level, you may get neurological symptoms at an older age, but with a lower enzyme activity as a child or an adolescent, that you're more prone to having retinal disease or pituitary disease. But that's very tough to prove. And when we looked at some of the different mechanisms, it was difficult to ascertain this in individual cells in culture because what we found was that PNPLA6 is expressed in a lot of different cell types. And so the ones in yellow are the highest expressing, the ones in green are moderate expressing, and the ones in purple are still expressing PNPLA6. So we see that it's expressed in the retinal ganglion cells, in the photoreceptors, in the RPE, and the choroid. So there wasn't really a great way of approaching this from a cellular perspective. Oh, and sorry, and I've lost I lost a slide there, and I'm sorry about that. But what I wanted to get to, let me stop sharing for 1 second and pull something up. What I wanted to share is some of our work that we were able to do in mouse, which helped to understand this activity of the PNPLA6 enzyme and the phenotypes. So 1 moment here. Just 1 slide. And so what we did was we created a mouse model. Sorry for my missing slide here. And bringing that back, but to show you this, that what we did was we developed a mouse model to better study PNPLA6 and the dysfunction. I hope you still see my slide there, but
No, Dr. Hufnagel, we can't see your slide.
oh, I have to click share. I'm sorry about that. There we go. Now it's okay.
Yeah, we can see, but it's not. We can see just an image of this topic trace and photopic trace.
It's perfect. That's great.
Yeah.
That's great. That's the slide I was missing. This is the content of that slide. Perfect. And so what we did was we generated a mouse model. And so the mouse model had 3 different genotypes. First, we had the wild type mouse. Then we had a mouse that had the spastic paraplegia mutation as homozygote. And then we had the spastic paraplegia mutation in combination with a null allele. And so our hypothesis is that if we can decrease the activity to a certain level, that we will elicit a retinal phenotype. And this is really the experiment that can demonstrate whether it's really enzyme level and not the diagnosis, the clinical diagnosis of the patient. And so what we did was we looked at this, we looked at the brain NTE activity of the mouse, and we showed that compared to wild type at 100%, that we can show a decrease in activity in the brain as a result of the genotype. So this is a mild missense genotype as homozygous, and this is a heterozygous missense with the frameshift null. And the reason we don't have a homozygous null is that this is lethal in mice. And in fact, we don't find people who have 0% activity either. And so, and then we looked at several things. First, we did an ERG, and we showed that the mice who have the severe allele have a decreased scotopic and photopic trace compared to those with the mild allele. We also found they have decreased total retinal thickness and outer nuclear layer thickness. And we also found they have a decreased optomotor response. So what this is telling us is that if you decrease the enzyme level of NTE far enough, you can develop retinopathy, but above a certain threshold, you're not likely to develop retinopathy. And so how do we use this then in the clinic? And how do we use this then in the clinic? And so what we're doing now is that we've tested many of these variants, and I'll go back to this slide here, and we've tested many of these variants, and now we have laboratories of people who have discovered patients who have PNPLA6 disease, and they come to us and they say, we have this variant, and we would like to know more about this, and do you think that this could explain the person's disease, and do you think that they have this particular clinical condition? What are the other things that we should look for? And we can say, if this person has these variants at a particular enzyme level, we think it's very important to be screening for vision changes. A lot of times people come to us and they haven't had any vision changes yet, or pituitary changes, particularly when they are in adolescence. So this is how we've done many of these genomic studies, and this has opened up a new area for us in terms of precision medicine for rare disease, where we're able to help patients know not just for diagnostic information, but also for prognostic information. And I just want to thank all the people in my lab who did this wonderful work, and thank you all for your attention. And apologies for my technical difficulties at the beginning.
Thank you, Dr. Hufnagel. This was really an amazing lecture. It actually shows a journey of how in-depth phenotyping as well as research has gone into to unveil the mechanisms behind this disease. And I'm sure there are many more diseases that you've worked on. It really shows the amount of work you and your students and your mentees have put into the understanding of these diseases and how we understand it today. So I particularly liked the way the disease, when they were described in the previous literature, I mean the older literature, and then they were matched to this based on the new learnings that we were seeing. So many times it happens that we cling on to the old nomenclature or old literature, but with new information coming on through genetics or through various other types of tests, we really need to revisit them and reclassify and recategorize these diseases. So thank
Absolutely.
you
I
for
think that's a wonderful comment. I completely agree.
Yeah, so that is what I really liked about this presentation. Apart from, of course, everything starting from first to the last slide, it was amazing. So I think similar to this is what why was ClinGen started, and that's what you work for. Can
That's
you?
right. In fact,
Yeah.
I did bring some additional slides for ClinGen, and one thing that you just pointed out, I think, is very important. The way that we describe things. So when we think about like even with RPE65 and gene therapy, you can see in the literature that there are multiple names for the RPE65 disease. We have LCA type 2, retinitis pigmentosa 20, and retinitis pigmentosa 87 with choroidal involvement. And so with ClinGen, we're also trying to make sure that we are properly assigning the names of diseases to those genes so that the clinicians and the insurance companies don't get confused when it comes to understanding what is RPE65 called and what does it cause. So now we're breaking RPE65 into the recessive form, RPE65 related recessive retinopathy, and RPE65 related dominant retinopathy, rather than using those old slightly more confusing terms. And now we've done this for, 100 different genes by now that we've gone through and really thought about what are the proper ways to talk about genes versus diseases so that we can really understand, the importance of, say, CNGA3 related retinopathy versus CAPN5 related vitreoretinopathy, and really understand the implications when it comes to therapies for these patients. Because if we use too many different names, it may create some confusion in who's eligible for a therapy versus versus having it available for everybody. And then this is just the structure of ClinGen. So if anybody's interested in learning more about ClinGen, I'm always happy to point you to any of our expert panels. We have a wonderful group of people. We have a wonderful group of people in ClinGen, and so I just put up these QR codes in case people want to contact me or contact the folks at ClinGen.
Yeah. So I'm sorry, I'll give other people also to speak, but I have I have a couple of questions more. So in your previous answer to the previous question, you mentioned about therapy. So why it is important for therapy. So I just want you to elaborate on that for everybody, that why is it important really to have this deep phenotyping as well as deep genotyping also, to know really, and what are the implications on therapy with some examples.
Yeah, I think with deep phenotyping, I think the part of the reason it's important is understanding the prognosis for the patient. And while we have very limited therapies, I think it is important to set expectations and also to intervene at the appropriate time. I think we're all well aware that sometimes if a patient has very little retina left, very little outer segment left, then a gene therapy may not be appropriate because the cells aren't actually there to respond to the therapy, in which case they may be better candidates for like a retinal prosthesis or similar. But I think it's even more important before we have therapies, because many of the clinical trials fail because the patient cohort does not meet the endpoints. And so we really need to have very good endpoints and probably combinatorial endpoints, so that we can really understand what does it mean to have success with a therapy. Because right now we're not reversing therapy or curing disease, for the most part. I think some of the CRISPR editing therapies are very interesting. When you do the actual gene editing, maybe that will stabilize disease, but still, to get it into so many cells in the retina and expect that disease is going to stop is, I think still premature for many of the therapies on the horizon. Our goal now is to slow disease as much as possible, and hopefully, as more medications become available, that they would be able to be used in combination to further slow disease, or at least slow them to the point where other therapeutic approaches would have more time to properly act. But I think this kind of deep phenotyping is critical both in the preclinical space, as well as post therapy. But for instance, for PNPLA6, because it affects all three layers of the retina, because we found that through deep phenotyping that it affects the retina, the RPE, and the choroid, it's very difficult to think about, a single injection in the subretinal space for a gene therapy that would really reverse the disease, and that it progresses so quickly is also very difficult because it is also very difficult because by the time the therapy has a chance to start working, the retina has already continued to degrade after that point. So you may not actually see a response. So that would be, one concern. But it is an enzyme, and enzymes are ultimately targetable. And so when we were able to show the different enzyme levels affecting causing retina versus pituitary phenotypes, I hope the same is true for therapy, that there may be small molecules that help to stabilize the PNPLA6 enzyme, and could those be delivered in a more general way, by drops or even orally, so that the PNPLA6 enzyme can be stabilized to the point where its activity is above that threshold for retinal disease, and maybe we can halt or at least slow progression of retinal disease for those patients. So I think in my view, a lot of the deep phenotyping has to do with which approach you want to use for each of these particular diseases, but it has to also be coupled, I think, with the deep biology to really understand like what your options are, and enzymes are ultimately great drug targets too.
Well, yeah, agreed. Yeah, thank you. Anyone else has a question for Dr. Hufnagel?
And I can stay late because I was late with my Zoom, and I am happy to stay late and talk to people as well.
Yeah, that's so kind of you. If there are questions, I have a couple of more questions, but yeah, Deepika, please go ahead.
Yeah,
So,
thank you.
Dr. Hufnagel, Deepika is one of our budding IRD specialists I should say, already an IRD specialist in
No, no.
in India. So,
Okay.
Deepika, go ahead.
Okay, thank you, Dr. Shailaja. I'll go into the question. Thank you, Dr. Hufnagel. It was amazing work and very good learning for all of us. My question is with respect to the enzyme levels in the retinopathy group. Like in the group where they had retinopathy and the enzyme levels were a certain levels, within the retinopathy group, was there any correlation between the severity of retinopathy, age of onset or age at which the retinopathy has occurred, and the varied spectrum of enzyme levels? Was there any
That's a great
observation on that?
That's a great question. So the short answer is we saw a trend for that. I can go back to that slide. We saw a trend for it, but it was not significantly different. We did try to look at, and I think part of it is because we're putting this enzyme exogenously into cells, and we're making a lot of it, and then we're doing a colorimetric assay, which is semi-quantitative, and we're comparing that to people who have complex genotypes. So these people have compound heterozygous variants, or most of them have compound heterozygous. They have two different missense alleles, and we're also relying on the literature. So I think since the literature is in a way semi-quantitative because we're using patient reports or researcher reports, when we looked at the age of onset of reported retinopathy and plotted that against the enzyme level, we saw a trend, but not a significant difference, because I think because of the semi-quantitative nature of the assay and issues with the accuracy in the literature, but we did see that the people who had the congenital retinopathy and congenital endocrinopathy did seem to be on the extreme end. So if we sort of took when it was affecting babies or early childhood below age 5, we did see that they tended to have very, very low enzyme levels.
Thank you. Thank you very much.
Shailaja,
We
can
also
I ask one
also
question?
Yes, ma'am. So,
Yeah.
Dr. Annie, please.
from excellent work. And what I learned from your lecture is that we should not give up the basics of genetics. Like if we are sure that it is a homozygous condition we are looking at, despite the fact that the gene test comes back with something autosomal dominant or something, we should pursue to look for the other variant or a gene which is showing a more relevant homozygous pattern. Because many of the reports which come, despite looking at the pedigree, which gives a particular genotype, which gives us a particular inheritance pattern, that is not documented in the genetic report. So now it gives me more hope to look more deeply into the genotype and see that it matches the hereditary pattern.
Yeah, and I agree. And I do think that it's best to try to reconcile that when you can. In some cases, we've also found that some patients sometimes it defies what you think it's going to be, no matter how we want it to make it all fit together. Sometimes people do end up with a dominant condition when we expect it to be recessive. But it's good to definitely turn over all the stones to try to find the answer. I would also say that in looking at the broader, large population studies, that also they find that between 1% and 2% of people who have rare diseases actually also have two rare diseases. And we found a number of instances where patients actually have two retinal dystrophies from two different genes, and that has happened. And there are case reports about that, of course. So sometimes we actually find one genetic disease on a person's report, but they may actually have a second genetic disease in the background. It's very rare, but it does happen.
Not very rare in our population where we have consanguinity.
Agreed. And I was going to bring that up, and so certainly for recessive disorders, and in consanguinity, it does increase dramatically. The likelihood of having a second disease goes up to almost 10%.
Thank you, Dr. Hassan.
Thank you
Dr.
for that.
Pawar, Dr. Neelam Pawar wants to ask a question.
Yeah,
Please unmute yourself, Dr. Neelam.
yeah. Really, it was a very wonderful lecture. And I had just one query whether in these cases any liver disorder, whether you have seen any, because other PNL, PA3 or 4, they are sometimes associated with liver diseases, but whether in particularly with this gene, whether any liver disorder, whether you have seen any.
No, and I think it's PNPLA2 or PNPLA3. And we worked with Pat Becerra, who is at the Eye Institute, who is also working on those genes and diseases. And it turns out that PNPLA6 and the other PNPLAs, they do have different structures. And they also have some have different substrates. So the PNPLA genes, they act in different parts of the cells, and they have different substrates and products. So they're very important phospholipases, but they have a variety of different functions in different parts of the body. And so far, we have not seen any liver phenotype or any sort of liver disease or liver cysts or anything else in our PNPLA6 patients. But we did look at that for the very reasons that you're mentioning. So that's a great question.
Yeah. Because considering the obesity in Lawrence Moon beetle and considering just like Wilson disease, last week only I saw Wilson disease. So like sometimes that liver disease and neurodegenerative things coexist. So just a query, like whether maybe in future, really we don't know.
Yeah, absolutely.
Thank you.
So, Dr. Hufnagel, one more question from my side. Are there any more questions from other people joining the webinar?
Hi, Dr. Shelja. This is
Yeah.
Dr. Anupriya. Can I ask one question?
Yeah, sure. But your voice is very feeble.
Yeah, am I audible?
Yeah, better.
Yeah, great work, Dr. Robert. Since you said you were associated with Clinton, I'm sure you must have heard, you must have read about that paper on how to grade a variant's pathogenicity based on the functional assay in literature. Those guidelines that have been given by Clinton. And what I remember after reading that paper is that those guidelines seem to be very stringent, that how to grade a variant's pathogenicity based on the functional assays that one has done into supporting pieces or moderate piece or a strong piece of evidence, how many times you have to replicate the study and all. So I was just wondering that when you people are doing these enzyme assays and all, do you take those factors that have been mentioned in that paper into consideration while you are
Question.
planning?
That's really great, and I'm sorry I forgot to mention that. So yeah, so we did proper replicates. We also did a number of 15 different benign variants. So that's another important part of that paper is that if you want to have a functional assay that is clinically applicable, you have to have both pathogenic variants and benign variants. So we tested 15 benign variants. We tested our almost 10 null variants, and then we tested, these 50 or so pathogenic missense variants. So we do reach this assay does reach the strong level in the ACMG variant classification rules by ClinGen for functional studies. So we can apply the PS3 strong criteria using this. And in fact, all the VUSs, variants of uncertain significance we talked about for the first three cases, those are all now pathogenic or likely pathogenic as a result of these studies. So thank you for bringing that full circle.
Thank you for answering.
Yeah, thank you. So, as a follow-up question, actually, I wanted to ask you that the first case that you showed, the type of genetic testing that had done was like a very old type of testing, right? So, how
So
often do you go back? Yeah, pardon?
Oh yeah, the first one was a
The first
research
case, really.
it was a research exome. So it was exome, but
Okay.
it was exome that was sort of done in the lab. So it wasn't a clinical grade exome. It was done by me. And so, it was
Oh, okay, okay. I thought it was some other
very early days.
early days. Yeah. So, at
Yeah.
that time, probably the exome was not readily available for clinical testing, was it?
That's right. This would have been in about this would have been like 2010, 2011 or
Okay.
so.
So how commonly now you go back and retest your patients or their samples based on the new technologies that come, and you go
Yeah,
back or just match them with the new variants that have been identified? I mean,
yeah,
in your
I think
practice, how often does it happen? Because you have such a vast experience, I just wanted to know out of curiosity.
Yeah, no, I mean, it's tough because I think generally speaking, the wisdom I keep hearing from other people is about every 5 years is not necessarily a new test, but you can also just reanalyze the data that was
Mm-hmm.
collected because the literature has changed.
Mm-hmm.
If there is an opportunity to go from like panel to exome to genome, each of those steps is about a 5% diagnostic yield. So panel to exome or exome to genome increases the yield, 3 to 5% each step.
Okay.
And then a reanalysis of the data is probably 1 to 2% change. It's not much, but it does happen because sometimes it's just the person looking at it, or it's the tools in the bioinformatic pipeline, or it can be the literature has changed. But there's many factors, and I think if you are going to go from like panel to exome or exome to genome, it's probably also good to use a different laboratory because then you get kind of fresh eyes on your patient because they wouldn't have been known to that lab already. But all labs are good. It's just sometimes good to shift from lab to lab if you're changing your testing.
Yeah, yeah, we have seen it in a couple of our patients also that when we get the same sample reanalyzed by different labs and we get different variants. It's just kind of confusing, but then when you understand that the pipeline may be different, all you have to do is then, correlate with the phenotype that you have.
Well, and so much of it has to do with the uh, uh, with the knowledge base that a lab has. So that each lab has like a database of variants that
Database,
they
yeah.
pull. They try to pull information from that rapidly because that's the easiest thing for them to put on the report.
Yeah.
But if you look at these numbers, like everybody differs from the reference by about 100,000 changes on the exome, and we have 20,000 genes, so that's about 5 variants on average per gene. So if you're doing a panel or you're looking for retinopathy across 300 genes, you're going to get 1,500 variants. And when we do our own panel testing, we see 1,500 variants, but almost all of them are known to be benign based on the knowledge bases. And so, you end up with only a small number of variants to really have to think about, do I put this on the report or not, if I don't find the obvious answer. And so I think that's the biggest thing. If you go from lab to lab to lab, you're going to see different VUSs. But hopefully they would pick out the pathogenic variants more easily because those are available on ClinVar, ClinGen, or others. But what I see from lab to lab is their VUS, their reporting of the variants of uncertain significance, and like different labs will report different sets of VUSs because they all have different thresholds for what they think is reportable.
Right. Okay. So, any further questions? No? Then great.
All right.
Yeah,
I
so
just want
thank
to
you.
put this up in case anybody. Oops. Sorry. In case anybody wants to contact me, I just want to put this back up. Yeah.
Sure, and you're on the group also, so I think people can contact you there also.
That's right. You've got my phone
Yeah.
number on WhatsApp. So yeah,
Yeah.
that'd
So
be lovely.
thank you so much, Dr. Hufnagel, and it was really, really amazing. And it's how you brought the rarest of rare on the Rare Disease Day for all of us. So it was ideal to call you for this lecture. And I think I'll request you to have more lectures for us if time permits and if it's possible for you. Especially, we would
Yeah,
want
of
to learn
course.
about the ClinGen and how we can more effectively use it or contribute to it. So
Yeah.
I think everybody would love to know that. And I think all of your case studies would be kind of examples for all of us to how to approach these cases. So anything you feel interesting, time to time, please to take lectures for us. Uh,
Yeah, will do. And you saw I had a set of of slides for ClinGen, but I talked too much about PNPLA6. So next time I'll give a full ClinGen lecture.
yeah, yeah, that would be
And if
great.
anybody will be at Arvo, we're gonna have a ClinGen meeting at Arvo.
Thank you, thank you. Not me this time, next year.
I know, I'm bummed. I'm sad that you will not be there.
Sure. Okay, thank you again. Have a great day. You can go back and sleep for a few hours if you want to. And
I
rest
think the
of us
kids are
also,
gonna
I
wake up now.
think.
I think my
Rest of
kids
us,
are waking up and your kids are going to bed. So
yes. My kid not in at least 2 hours because it's
nice.
Saturday, so it's late night for him.
Right.
But I'm sure many of us are past their sleeping time and dinner time here. So thank you everyone who joined. It was really great, and this lecture would is recorded and will be